to introduce here Valentina, Carl, and Yasmin, who have volunteered to give an introduction into Exomiser, which is one of the flagship products in the kind of wider Monarch sphere that many of us are involved with, work with, but actually many of us have never like gotten any hands-on experience with. So thank you very much for you three to agree to do this, and yeah, take it away. And I'm going to close my video as well. So yeah, thank you very much.
Right, so I'm going to share some slides. If I can do it effectively. Okay, maybe I can put this down. So can you see my slides?
Yes,
Maybe
we can.
I can put them in this presentation mode.
Perfect.
Right. Okay, so hello everyone. Welcome to this tutorial, an introduction to Exomiser. I'm Valentina Cipriani, and I'm here with Yasmin Bridges and Carlo Kroll. We are all from Queen Mary University of London, and Damian Smedley and Jules Jacobsen online too. So the main developer of Exomiser. So that's good. As you know, this tutorial will be hands-on, so you will be running a simple analysis with us. But we also thought that we could give you a very brief overview on Exomiser, just to be on the same page as much as possible. If you're here, maybe you know already that Exomiser is a so-called variant prioritization tool that works for whole exome sequencing, but also for whole genome sequencing. This tutorial will focus on whole exome data. Probably we are implicitly volunteering for another tutorial on Genomiser, so to analyze the genome data. It's a tool that can assist molecular diagnostics, but also novel gene discovery in the field of rare Mendelian disease. So it's tailored to Mendelian disease, and it wouldn't work for complex genetics, for example, not strictly speaking. Right, so why do we need such tools? When sequencing arrived in the field, I think that the hope was that we could analyze a VCF file and apply a certain standard variant-based filtering criteria that could get to a single disease-causing variant. For example, we would get rid of bad quality variants, common variants using publicly available data sets, or also in-house sequencing data set to get rid of artifacts. We would focus on protein coding variants only. We would score the variant with some in silico predictive pathogenicity algorithms. And if we were lucky, we will also have access to some other information, like, for example, inferred mode of inheritance, or previous linkage and IBD studies to narrow the genomic searches. As well as, for example, sequencing data from multiple affected and unaffected family members, or even sequencing data from multiple families. But the reality, it's quite different, meaning that no matter how strict these filtering criteria based on variants are, what we get usually, it's really more like this cloud of potential candidate variants. They are all pretty rare based on our criteria, and they are all pretty predicted to be pathogenic. So usually these variant-based filtering criteria are not very highly discriminatory. And again, if we are lucky, we may identify in this cloud of variants previously reported variants in genes that have been previously associated with a disease or a similar disease, or clearly pathogenic variants. So this can be solved in an easier way. But usually we also get a lot of variants of unknown significance, so-called VUS, in genes that have been previously associated with the disease, or the most general case, variants in genes that have not been previously associated with the disease. So clearly, this is not satisfactory. And what Exomiser does, together with actually other tools now, is that it brings in the phenotype in the actual analysis. So diseases are a set of phenotypes, so they can be encoded using, for example, the human phenotype ontology terms. And this way we can make direct use of the phenotypic information in our variant prioritization exercise. So in a nutshell, and obviously we will look at the actual analysis and the output files later, Exomiser works, I would say, in 3 steps. So the first one is the most standard steps, and it would take as an input a VCF file. It can be a single sample, but it can also be multi-sample, so family-based with some pedigree information. And it can come from whole exome, but as I said, also from whole genome. And we would apply essentially the criteria that I mentioned before, also within Exomiser. So we can focus on rare variants only, protein coding variants only, and we can apply mode of inheritance, select some in silico pathogenicity algorithms. This will be all clear when we look at the actual analysis settings. So from this step, we would get a set of filtered rare variants that receive a variant score that goes from 0 to 1. So 1 is the maximum variant score, meaning that variant is rare enough based on our criteria and is also pathogenic. The second step is the phenotype matching and gene scoring. And that's the new element in comparison with other tools based on variants only. For this step, Exomiser needs the phenotypic profile of the patient encoded with a human phenotype ontology terms. And it makes use of an algorithm called PhenoDiagram. You can find all these references in the Exomiser docs. And it will give a so-called phenotype score that goes from 0 to 1, where 1 is the maximum, per each gene. Essentially what it does, again in a nutshell, and we will look at the actual result, it takes the phenotypic profile of the patients, and it looks for any phenotypic association, any phenotypic profile in mouse model or in human diseases, or it will also look at the protein-protein interaction data, and it will tell us if our patient phenotypic profile is similar to any of these phenotypic profiles in the databases. For example, we may have a patient affected by the Pfeiffer syndrome described by this set of terms. Exomiser may find some rare candidate variants in the gene FGFR2, and then it goes to the mouse model database. If a phenotypic annotation for the ortholog of this gene exists, then this set of terms that for the mouse are encoded in a mammalian phenotype ontology are compared pairwise with the set of terms that we give as an input. And it's essentially a semantic similarity. So the score represents, in essence, how much this set of terms are similar to the set of terms that describe the phenotype in the mouse. And similarly, this can be done also between the set of HPO terms of the patient and the actual human phenotype ontology terms from OMIM, for example, for any disease associated to the gene of interest, and also for the zebrafish model database. The other important phenotypic match is the one that exploits the protein-protein interaction data. And for example, we may have a patient described with this set of HPO terms on the left-hand side. There may be some rare candidate variants in, say, gene MED23. Exomiser retrieves from the database strings all high confidence interaction with this gene. And let's suppose that, for example, in this gene network, there is a second gene, NIPBL, that is annotated with these HPO terms as associated to the disease that is named here. And therefore, again, the comparison is made and the phenotype score is produced. This type of comparison is particularly helpful when, for example, there are no non-human disease for the gene of interest or no other animal model. So it's very helpful for discovery, for example. So this means that we have a variant score per each variant and a phenotype score per each gene. And the two scores are then combined in what we call the Exomiser combined score using this formula. You can find again all the details in our papers in the references. So by doing this, what we are hoping is that instead of looking at the cloud of variants where the pathogenicity is close to each other, we are hoping that the ranking is more discriminatory, and so we can go from the best candidate to the worst candidates in a more effective way. So that's in a nutshell, because really we don't want to spend too much time on the theory, and we will now go to the actual tutorial. I shared again the address in the chat. So we hope that if you are here today, you went through the prerequisites. So you want Docker installed and a few databases downloaded. But it's not harmful to open the terminal again and to actually do a Docker pull. You should see that your image is up to date if you install Exomiser via Docker already. So while you may want to try this, I want to mention that Exomiser can be installed also via Windows, and we left some instructions here. I tried the instructions many times, also this morning on my Windows laptop, and they work just fine. So hopefully it will be the same for you if you choose this route, and if you watch the tutorial at a different time than now. And of course, it can be installed also via Linux or Mac, so through a terminal. And again, we left all these instructions that should work just fine. I can show you probably how it looks like under our cluster. That should be already open. So this is the Queen Mary cluster. So we have actually many releases of Exomiser, but for this tutorial, we are going to use the latest one, which is 13.2.0. And when you download the distribution, the folder will look like this. And in the examples, there will be essentially most of the analysis files that we share with you today using the Google Drive. So this is only if you go through a different installation. But for today, again, you should have followed our prerequisites. Right, so now I think, so I have a tight lineup here. So we have Carlo that will show you how to configure the application properties file. So I'll stop sharing.
Okay. Right, so we start with the application properties file, but before we wanted to actually start with the Docker command.
Right, yes, so
Okay.
I mentioned the Docker. Do we know if
Okay.
anyone had
No,
issues? I think that's that should be all done.
So in the beginning, did anybody had issues with the Docker pull command, or was it working for everyone?
It was working for me for sure, so I just want to confirm.
Yeah, then I just assume that everybody's fine.
Mm-hmm. And
Okay.
there are no other questions in the chat. So,
Okay, right.
okay.
So to actually run Exomiser, we always need like a configuration file, which in oh, I should share it.
Yeah.
Okay, we have this configuration file, which is basically there to point the data to the right location where it needs to be for the Exomiser. And the simplest and minimalistic setup for this whole file is where you just need to create those 3 lines to set the data version, like we use 2302 data, and here to set the file path to where it is. So right now we're using an Exomiser data file path because we're using the Docker installation, and we're later mounting the directory where it actually is, like in our Exomiser tutorial directory to the Docker. That's why we just have the /Exomiser data here. To those things here, to the RAM and CAT version, there's additional databases which can be used, especially RAM is used for the Genomizer. But in this tutorial, we won't need that. If we want to activate this will be done like in those lines. And additionally, we will have to configure the whitelist from ClinVar, which is in this line, so that we don't have the problem that we already analyzed variants, and ClinVar has already set them to a high score of pathogenicity, so that it's like safe that it's already done, right? And we also have to state the data version for the phenotype data, which in this case is also 2302 data. Okay.
Right, and that is all done for you. So we provided this application properties already set up. So now I think that we can actually go to the running of Exomiser, so the real deal.
Yeah.
And we will have first Yasmin that will explain the data and the phenopacket. Then I will go through the analysis settings. And with Carlo, you will go through the actual command via Docker. So maybe, Yasmin, you can share.
So for this tutorial, we'll be focusing on running Exomiser on a single sample whole exome VCF. So this sample is not based on real clinical data, it's a simulated data where a known variant for Pfeiffer syndrome has been spiked into a normal VCF from the 1000 Genomes Project. So the recommended input for Exomiser is a phenopacket, and these can be accepted in either YAML or JSON format, and it can also deal with version 1 and version 2 phenopackets. But this sample here for reference is YAML format and is a version 1 phenopacket. So I won't go into too much detail about phenopackets because there's been a previous Monarch tutorial by Jules on phenopackets, but I will go over what the bare minimum is to run Exomiser. So you will need an ID, and this is an arbitrary ID, so it can be whatever you want it to be, but in this case, it is the same as the proband ID. And then you have the subject field, which describes the individual that we're looking at in the sample. So it can describe the gender, the date of birth, and as well you need this ID, which Exomiser uses to look for in the VCF. And then you also have this phenotypic features element, which is intended to describe a phenotype that categorizes the subject of the phenopacket. And this is also required, and this is where you have the HPO IDs from the human phenotype ontology, and you describe it all in this element here. It's also recommended to have the files described as well in the HTS files element. So this is any files that are associated with the proband itself. So you can add the link, the path to where the VCF is located, and Exomiser will read it from here, and as well as the genome assembly, so it knows which database to use. The metadata isn't necessarily used by Exomiser, but it is a required field within the phenopacket schema. And it's just to describe any resources used to create the phenopacket, as well as having some references to which version or which ontology you use to describe any of the elements in the phenopacket. So there are many more elements you can add to a phenopacket, but I won't go into detail, but I have added links to the phenopacket 2 schema, as well as how you can convert this to a version 2, which tools. So now Valentina will go over the analysis settings you can use with Exomiser.
Right, thank you. I'll share again. Right, so you also received in the Google Drive or otherwise in the repo a file called Pfeiffer analysis, again in YML, and it's ready to be run, and we use most of the default settings. But if we go through the settings now, I'd like to mention a few more things so that you can have some more flexibility if you run the analysis again on your own. So first of all, you can specify whether you want the full analysis or pass only. We would recommend to use pass only because otherwise you would get all the possible variants. So with pass only, you get only the filtered variants. But if you are doing any benchmarking and you have, for example, you are after a specific variant that may get filtered out, maybe you want to try the full analysis as well. So the first option is this inheritance mode. So here you can specify both any infer mode of inheritance if it's known, and the corresponding frequency filtering. So these are our suggested cutoffs. Obviously, you can change them. It's important that when it comes to recessive, either autosomal or X-recessive, you need to specify both frequency cutoff. So the first one is for the compound heterozygotes, and the second one is for the homozygotes. Similarly for X-recessive. Then the second option is these frequency sources. So here you specify the actual sources for allele frequencies. I would say again, don't touch anything, but you can see that here we use the usual, so 1000 genomes, .med, UK10K, EXAC, but also nomads, the exomes, and nomad, the genomes. Most of this data, the previous data set, probably overlap with nomad, but if you leave this, all these details, you will also receive the corresponding details in the output file. So I would say you can leave this as it is. In terms of the pathogenicity sources, so these are the possible options available within Exomiser, so polyphen, mutation tester, CIFS, Revel, MVP, CAD and REM if they are activated in the application properties, and REM is specifically for genome data. Our recommendation is to use Revel and MVP. This comes from some previous benchmarking on simulated and real patient data. Then you have a series of steps that actually they are all not mandatory in a sense. So here we highlighted the default one that we want to use today. So failed variant filter, if it's activated this way, it means that it will filter out variants that don't pass the filter within the VCF. so that they have a non-pass value within the VCF. So you want to trust your bioinformatics pipeline. Variant effect filter essentially is if you want to run specifically a whole exome analysis. So if you use this, if you remove these labels, it means that you are focusing essentially on protein coding variants only. These labels come from Jannovar, which is the software used internally in Exomiser to perform variant annotation. If you are running a genome analysis, then you want to include some or all of these category essentially, and you won't remove them. But again, today it's about whole exome data. The frequency filter, I don't know if this will stay also in future releases, but essentially it's the maximum frequency that you specify up in the settings that I showed you before here, the inheritance modes. So for us, it's 2%. Let me go down quickly again. This is probably also something that we use maybe back in the day, this pathogenicity filter. So keep non-pathogenic by default, we want all of them, because otherwise we are saying that if a variant is not predicted to be pathogenic, it gets filtered out. But we know that these algorithms have their own limitation. Inheritance filter and noming prioritizer, as it says in the comment here, we try to leave, by the way, all comments, as many comments as possible. Obviously, when you run your own analysis, you may want to have a cleaner analysis settings files. So inheritance filter and noming prioritizer should always run after all other filters have completed. So at this point, so here we are actually applying the mode of inheritance and frequency filtering. And the oming prioritizer is not mandatory, but we suggested to use it. It will be clearer when we look at the output files. Essentially, if we have a variant in a gene which is associated with a non-human disease with a certain mode of inheritance, but in our sample, the mode of inheritance is different, the prioritization will receive a penalty if we apply this line. The high-fi prioritizer is the actual moment, the actual step when Exomiser produces the phenotype scores. The name is from the original paper, the 2014 Robinson paper. 5 is an acronym, you can check the reference. And if you use it this way without specifying anything, it will make use of the mouse data, the human disease data, the fish, zebrafish data, and the protein-protein interaction data. For benchmarking reason, you may want to specify with this parameter, for example, only mouse or only fish and so on. But again, you will leave it this way. Then that is all essentially. And now we have at the bottom these output options. Again, I wouldn't change much of this. So output contributing variance only is set to false. This will be clear when we look at the actual output files, but essentially within each gene, you may have multiple filtered variants that pass all the criteria, and Exomiser will pick the best one to contribute to the final score. But in the output, you still want to see these multiple variants. So this is set to false. Num genes set to 0, so you get all the genes as opposed to a limited list. And now this is recent. You can specify the actual name for the output directory, that in our case will be results. The actual prefix of all output files. And in terms of the output formats, these are the all possible formats. So HTML, JSON, TSV gene, TSV variant, and VCF. By default, I believe maybe this is a minor a typo. By default, we can get both the HTML and the JSON file. In our tutorial, we are producing HTML, JSON, tab separated genes and variants. And obviously, we will look at these files in detail very soon. So that is all in terms of analysis settings. And now I believe if Carlos shares his screen, you can run the actual analysis.
Okay. Before actually going over the real command and everything, I just want to make a short summary of how your Exomiser tutorial directory should look like. So you have your Exomiser config, Exomiser data, and the Exomiser overview. Okay, that's not important for now, but the Exomiser results directory inside. And for me, it's like located on my desktop. So I just run the command now, but we will go through it, especially from here, from this part. So if we break it down, let's start with the first command. The docker run command will basically just tell the Docker container to run. And then the IT flag is kind of important because it opens the container in an interactive mode with the terminal attached, so that we can basically afterwards mount data from our local machine to the container, and that we can use it and then later also grab the results back to our local machine if we would like to have this. And then we have those 3 -v flags, which then on the left side, separated by a colon, just has the path to your data, or like in this case to the Exomiser data on your local machine. And then it will create this directory inside the Docker container. The same goes for the Exomiser config with all the example files, and it will create the Exomiser directory inside the container, which we then can use with the Docker commands here, downside here, where we can then say, so when we actually run the Docker with Exomiser and like take the image and then do the sample and analysis commands, we would not go to the local machine and ask there for the data. We would go to the Docker and ask there for the data. So in the Exomiser directory where the data is mounted to. And then in the end, we would just have to add the application properties file, which is also mounted to the Docker container, to make sure that the analysis is going the right way and using the right data. So when we actually run everything, you can use those backslashes to put everything into the terminal. I just put everything behind each other for this tutorial here. And as you can see, like, for example, I showed it before, like this is the path to my Exomiser data directory, and it never changes. And then I'm just mounting it to another directory inside the Docker. So when the analysis is actually run, you will see inside your Exomiser results, those 4 result files. And because we in the analysis file specified only, so everything without VCF, and that's the part with this command. Right.
Right. So we also provided an extra command if you want to run Exomiser locally. I mean, if you went for the local installation, but we won't do it live today. And so hopefully people are following along and they try this command now, and they obtain those 4 output files as Carlo described. If there are any burning issues, you can paste it in the chat. Otherwise, we can accept the questions at the end.
Yeah. One thing to add is maybe that we also have another Docker image which has a bash shell incorporated, so that if anybody wants to run like bigger pipelines, for example, with Nextflow, you would more likely to use this. But for this tutorial, we don't need really a bash shell, so we haven't used it.
Right. So now we want to mention also how to run multi-sample analysis and large analysis. Again, it's not something that we will do live with you today, but we left all the instructions detailed in the Exomiser tutorial document. So, Jasmine, do you want to take care of the multi-sample analysis?
So you can also run Exomiser on multi-sample VCFs. So these are cases that we have variants from multiple samples from one family that are contained in a single VCF file. And as well, you would need the pedigree information for this. So you can see that the phenopacket structure is slightly different to the single sample VCF in the terms that the ID has changed, where it's the family ID. And as well, we have this pedigree information that's detailed within this pedigree element. So this is where you point to for each individual in the family, you have the individual ID and the paternal and the maternal ID, as well as the gender and the affected status. And you can see that the VCF file is pointing now to this quartet VCF, which obviously has the variant information for these 4 members of the family. So we've provided the Docker and the local commands if you want to try it in your own time. Carla will now go over how to run large jobs.
Right, to run larger jobs, you can actually use batch files. So we have one batch file also in the Exomiser config, which you can find under Exomiser batch files right here. And this one looks like that. And you can basically list a variety of commands there, which then will be run by Exomiser. So, and it's faster than just doing it one by one, and it's also better for the CPU of your computer. And also note that if you're using Docker, always take the file path to the directory inside the Docker, because from there it's where the Exomiser is run from, and then you will not have problems with data versions or anything else and getting some errors. Yeah, that's just very fast for that.
Thank you. Right, so now we reached probably the most important part. So assuming that you were able to run the analysis with us and you obtain all those 4 output files, we want to comment as much as possible, I mean, within the time allocated, the HTML file,
yeah.
and then we'll go to the JSON and the tab separated
right.
file.
So that's our HTML results file, which is created by default together with the JSON. And it's separated in, let's say, 4 parts, which you can see here in the top, or 5 parts, sorry. And this, where just basically the first 3 parts are giving like a summary of what we've already done. So it gives you the analysis settings again. I won't go deeper into that because we already did that. And then here it gives you also a filtering summary. So in that case, the first 3 or the first 4 summaries are variant filters, which have been activated for this kind of analysis. And we only use past filters. That's why we don't we only use the past analysis mode. That's why we don't have failed filters. And as you can see in the bottom here, this is a gene filter, and that's why we possibly have a little bit less, a lower number there than in the others, because of course there can be more than 1 variant in a gene. And also we will have a variant type distribution scale, where you can see which kind of variants actually are in your analysis. And I just want to show you a quick example how it would look like if you do it in full analysis mode. We don't recommend this because it takes a lot of time. And yeah, as you can see, then you will have like larger numbers. And but just to show that shortly. Yeah. Okay, now Valentina will go a little bit deeper into the gene faction.
Yes. Right. Then, so I'm sharing, right? So I would say the most relevant part is the prioritized genes. So I want to show you an overview, and then I've got 3 screenshots. So you will have the actual list of prioritized genes and variants. So if I scroll down, you will see other genes appearing. This is really a dense output file. It contains a lot of active links. And I would say it's perfect when you want to share the results, for example, also with the clinicians, with other scientists. So in terms of the screenshot that I took, I will focus on the first gene in the list, which is, surprise, surprise, the causative gene, but because we know that we spike in the actual variant. So that's the first screenshot, right. So what do you have in this prioritized genes section? It's a list of candidate genes and variants sorted by descending Exomiser score. So in the header, after the name of the actual gene, you will have the variant, phenotype, and combined Exomiser scores. It goes with the highest scoring compatible mode of inheritance, if more than one. So in this case, this gene is, I would say, prioritized with a very high Exomiser score, also with a very high phenotype score, and with the maximum variant score. So we'll take a look at all these details. There is also a p-value. This is a recent addition that comes from bootstrapping, and it's described in the tutorial. You can look at the detail on how it was obtained. I think this is, in a sense, still a work in progress, so it may change in future releases. Then you have the section called phenotype matches. So here you have the best human disease model organisms and protein-protein interaction evidence for this gene, if they exist. So for example, for the human disease evidence, Exomiser highlights that there is the upper syndrome associated with FGFR2. So this is from OMIM. This disease is phenotypically annotated with the list of HPO terms that you see on the right-hand side. And you can also see how the patient HPO terms provided as input matches with the one from the human disease. And the phenotype score, which is called here phenotypic similarity, is 0.867. Actually, the best phenotype score, which is then the one that contributes to the combined score, comes from the mouse model data. So there is an ortholog for this gene, and in the mouse, the phenotype that you can see described with the NP terms here on the right-hand side is observed. In this case, we also have some evidence from the protein interaction. It's called proximity scores, but it's not the best score. So Exomiser takes the best of all possible phenotypic scores, whether from human disease, from mouse models, zebrafish, or for the protein-protein interaction. In this case, sorry, I didn't say for the protein-protein interaction, the second gene is FGF18, which is associated with actually this mouse phenotype. So the protein-protein interaction can look at, again, any human disease, any mouse model or zebrafish model associated with the second gene in the network. The other screenshot I wanted to show you is that is part 2. So in this case, also a long list of non-human disease exists, so this part can appear or not. And you will have all the OMIM codes. These are all active links, and also ORFA codes if they exist, and the specification of the mode of inheritance. The third screenshot is this one. This is also quite dense. At this point, you would have the detail of gene scores under each compatible inheritance mode. So, for example, under autosomal dominant, you will have again all the scores, and more in detail all disease matches and phenotypic similarity for this mode of inheritance. So this is it comes from the list that I showed you a second ago. And the last part is the variance section. So these are the variants actually contributing to the score. So for each variant or variance, if it's compound heterozygote, you will have the consequence. So this comes from Janova. In this case, it's a missense variant. The type of variant, so SNV or Indel, and actually now Exomiser can also analyze CMV, chromosome, position, ref and alt allele, the genotype, and any RSID if it exists. The Exomiser ACMG annotation is relatively new. You can find all the details in the Exomiser docs. It's a subset of all the criteria, so it can be used to flag the evidence more and to assist the analysis, but it shouldn't be like the final ACMG annotation that can be done with, I mean, more manually with clinicians in a multidisciplinary team way. In this case, this variant is also reported in ClinVar as pathogenic or likely pathogenic. So you would have these fields if that's the case. And the variance score, again, it's equal to 1, so to the maximum, precisely because this variant is previously reported in ClinVar, and actually it's present in the white list of Exomiser. So this means that whatever the variants, whatever the pathogenicity score the variance receives, actually by default it goes to 1. Otherwise, Revel and MVP would have scored this variant slightly less than 1, as you can see here. You can also have all the transcript annotations. And if there is any frequency data available, not in this case, it will be documented, it will be detailed here. And I think that's all I wanted to say. So obviously it's very dense, but now let's move to Jason and we'll finish off with the TSV output files.
So the JSON represents the most accurate representation of the results as it's referenced internally by Exomiser. So you can use Python or JQ to extract data from this file, and I provided some simple Python codes to how you can iterate over the data and extract relevant parts. So Carla and I will show you how the JSON is visualized and where you can find some relevant data that you might find interesting. So here it's arranged in an array of these predicted genes. So you can see we've got 228, which is what we've got from the HTML. So when we go into it deeper, you have the gene symbol, so this is the predicted gene, and then you have all the relevant gene identifiers for this gene and the combined priority, variant, and p-value score. So these are the best scores for the mode of inheritance. So if you go into the gene scores here, you have all of the modes of inheritance, so autosomal dominant, autosomal recessive, and all the rest. And you can go in here for the contributing variants, and here you have all the information for any of the variants that Exomiser has predicted for that specific gene. So now I'll hand over to Carlo.
Yeah. Additionally, you will have the priority results sector, and you will find like the different results for the high 5 priority prioritization of genes and the OMA priority. And especially, which is like very important and linked to the HTML results, will be the disease matches part, because here you can basically see which diseases are matched to the gene, and then you can go to the best model phenotype matches, and this will be like the matches in the end which were shown in the HTML file. So basically, yeah, those things. And it's always the best match which is given in the HTML, but in the JSON you will have all the different scores and everything which is related to it. And we basically use a combination of the IC score and the SimYard score to rank those. And just to make a short description to this, these are both measurements to use and quantify the semantic similarity between terms in ontology-based data. And yeah, both are using like different algorithms to quantify this, and we combine it then to prioritize the genes, right?
Right, and then the last output file is the TSV. So let me share again. So we asked for both dot genes and dot variants TSV files. So I can open the, for example, the dot variants. You will find many columns here. I would say most of them are really self-explanatory. Probably an important observation is that you can see here, for example, that a gene can appear multiple times, and as well, for example, the same variant can appear multiple times. So I'm highlighting these two rows. So this is because MUC6 is ranked 7 under mode of inheritance, under dominant mode of inheritance, and is ranked 8 under recessive mode of inheritance. And for example, for the dominant one, we have this variant in row 8, and together with the different variants, this is selected as compounded heterozygote. And another observation is that you can have, because of the settings that I showed you before, you can have multiple variants within the same mode of inheritance displayed here in the output file, but only one of them will be the contributing one. In this case, the one that I'm highlighting here. So under contributing variant, you can see that it's got value 1. And that's because the variant score is slightly better than the other one. Other fields, as I said, are really quite self-explanatory. You can reach out if you don't understand some of them. But it's essentially a very good summary also of what we saw in the HTML file. The other file, .genes, and that's the last thing we want to comment, show a prioritized list of genes, so without the variant information. And again, also here you can have, for example, a gene that can appear multiple times. So again, this MUC6 under dominant or recessive mode of inheritance. And what is interesting here is that you have all the different phenotypic scores, and also the actual HPO term matching. And I mentioned the OMIM prioritizer in the analysis settings. So if we can see the values, sometimes it's not equal to 1. So these are examples of diseases where, for example, if we take this gene, in our patient is prioritized under mode of inheritance dominant, but if we go to OMIM, you will see, if you search for this ID, is actually a recessive gene. And that's why internally Exomiser gives this sort of penalty. That's really all I wanted to say, also because we are approaching the hour. But I hope we have still time for some questions. Nico, can we take a few questions?
Absolutely. Before we take questions, I just want to say this was absolutely awesome. And I think that many generations of people growing in Monarch will use this tutorial in onboarding, and we will go through it frame by frame, because it was indeed dense in certain parts and really dissect everything. So this was great work. Thank you very much. And I hope that I speak on behalf of everyone here.